Now I want to shift to scoring of the entries. We are going to be using multiple converging metrics for the scores, and we'll provide you details and rankings on the scores. So let me begin with the multiple converging metrics issue. There is no gold standard for connectome data set for any animal, be that human or primate within that set. There is substantial data on animal systems, for example, work by Van Ness and Young and Hellman, and some human works by Munz and Hageman. These are people who, some of these are on our board. However, even though we don't know the complete answer, we do know features of the brain that will surely be part of the correct answer. For example, the major borders, for example, V1 and V2, are widely replicated across species, individuals, and replications within an individual. If you get the right connectome map, you should be able to find those borders, and hence that's part of our test set. Major fiber tracts, for example, the corticospinal tract, are widely replicated again across species, individuals, and replications, so we can use that as a metric. The topology is unknown, but the basic topology is likely to be reliable within subject and close similarity between humans, and we'll be looking at the replicability of the topology of the methods involved. We will be using multiple metrics, in essence 3 categories of metrics: area metrics, matching the functionally defined areas of brain areas, V1, V2 for example, fiber metrics to match coded fiber pathways, and topological metrics between subject reliability of the subject topology at a given scale. The total score metric will be a weighted average of these individual metrics. So to illustrate, when the competition is done, you will get a set of scores on how you did on each metric, and perhaps yours would be the black line here for very good performance within that set, and what the best performer was on each metric within the competition. So let me give you again the example of the overlap test. You'd identify V1 and V2 based on the diffusion weighted and resting state data, as illustrated by this map. Then your technique would draw the borders in and label that on the surface as to what is area V1 and V2 within that process. And then we would calculate a tentative score based on the standard, which we will have only in Pittsburgh for subjects 2 through 4, and your own data within that process to give you a metric of the degree to which you match the overlap of some known cortical areas which are defined by our functional mapping techniques. In terms of the fiber tracking, this is an example where the red and green blobs here are the hand areas for this individual, and then we have the mapping of the fiber tracks that the individual should have gotten coming out of the hand areas within that set. Then we will compare now the boundaries of the fiber tracks, where they ended up from a source to destination, to see whether or not your technique found the targets from the regions which were initially specified with both human and functional mapping of the areas in question. And again, we will calculate a quantitative overlap score for the degree to which you matched known pieces of the fiber system. A way to think of this is like a standard intelligence test. We are sampling particular pieces of knowledge of the connectome and giving you a score based on our sample. You will not know beforehand all of the samples that we might use. Now we turn to topology metrics. This is still being determined. Entrants will be asked to create a connectome topology that produces a map for the 4 individuals based on the regions and the overlap topology and the functionally identified areas. Some suggested metrics that we're currently in discussion is between subject cross-connection metrics of the aligned areas, that is the correlation of the connection matrix from one individual to another, measures of similarity of the elements. For example, there are techniques that have been developed for measuring networks on the internet that give us a measure of the degree to which a network is similar from one point in time to another. And there are analysis techniques that look, for example, at hubs and modularity of the brain within that set. And we'll create a set of these metrics. We will do this both for the full brain and for subsystems such as in vision, audition, motor, language, and cognitive control net. We will provide a combined metric. We need to have a single winner within this process. So we'll provide an aggregate score which will determine who is the winner of the cash prizes. The weighting between factors is proposed to be the average of each category. We may use an aggregate score based on z-scores within a given feature. Note, we are likely to have a criterion that in order to be a winner, you must score in the upper proportion, top 10%, top 10 entries on the area and track metrics to qualify for prize consideration. We view these as validity checks for the topology method, and you must score at the high level on the validity tests before you can consider in combination with the topology scores for the prize. So here's a simulation that illustrates the kind of score that you might get in June, where we'd identify for each of the individual features what your score was, illustrated perhaps here by the black line, and the scores for each feature. This, by the way, is an example from the data of the 2006 competition where I just took the scores. Note, you can see on each feature if you did well relative to others. And one of the things that we found very pleasing in the previous competitions, that people who did well on some features but poorly on others read very carefully the features where they did poorly. And then next year, when we did the competition a second time, they utilized the other techniques to do well. You will also get your relevant ranking relative to all the entrants within the competition. I encourage you to participate in the forum and discussion. We solicit input for the scoring metrics, the aggregate methods on the forum. There will be threads for each of these metrics. And please, if you think you have a better idea, please post them on the forum or email us so that we can consider those. The board will consider suggestions, and specifics will be released in the guidebook and in the next webcast. Now let me shift to the data that will be submitted by the researchers. The first, in essence, think of it as you will be submitting data which gives the segmentation of the brain areas and the fiber tracks of the brain as illustrated in these two images. We will provide the initial masks, and then you will fill in those data. So for the mask, for each area, the areas, the cortical areas identified here in red, you would identify what your designation is of that cortical area in terms of some sequential number, and for each white matter area, the fibers that you believe to be passing through that area, with a maximum of no more than 5 fibers in a given voxel within that process. We'll provide a full brain cortex map of the cortical mantle and the subcortical areas, and then you'll code the cortical areas and the subcortical areas. Provide labeling of the fiber pathways. We're not concerned about the name you give that label, but that it is a coherent pathway from one area to the next within that system. And then entrants will be provided a full brain volume segmented for the gray matter, white matter, and other areas to put these labels in.